What is gene therapy and cell therapy?
Therapy is a medical strategy that addresses the underlying genetic issue in order to treat or prevent disease. Instead of utilizing drugs or surgery, gene therapy procedures allow doctors to treat a problem by changing a person's genetic composition. It entails the delivery of genetic material, typically in the form of a carrier or a vector, and the incorporation of the gene into the proper bodily cells. The initial form of gene therapy, also known as gene transfer or gene addition, was created to add a new gene to cells to aid in the fight against disease and a healthy copy of a gene to replace the disease-causing mutated copy.
Cell therapy involves introducing cells into the patient with appropriate function. The use of human cells in cellular therapy involves replacing or mending harmed cells or tissue. An example would be blood transfusion and stem cell therapy, in which it is simply one of the cutting-edge approaches employed in a variety of diseases. Many different types of cells may be used as a part of therapy or treatment for a number of illnesses and disorders thanks to new technologies, inventive goods, and infinite imagination.
Gene therapy works by inserting a normal gene into the genome in order to replace an abnormal gene responsible for a particular disease. One of the most significant difficulties with this treatment modality is releasing the gene into the cell. Therefore, a molecular carrier termed vector is employed to release the gene. Viruses are often employed as vectors where they are administered intravenously or injected into specific target tissues. The virus then infects surrounding cells in the tissue and delivers the gene embedded in it.
Now, let us know what are the strategies in gene therapy. So the first one is the gene addition. It is where it targets a faulty gene within a patient's cell by adding a functional copy of that gene to affect protein expression or by inserting a different gene that circumvents the defect and restores cell function. The second one is the gene correction. It is one of the mechanisms of gene editing and gives researchers a powerful tool for both repairing and updating DNA, for discovering gene functions, and for engineering new genetic variants. The next one is the gene silencing. It is an epigenetic modification of gene expression leading to inactivation of previously active individual genes or larger chromosome regions.
Reprogramming is transforming already mature and specialized cells into induced pluripotent stem cells or iPS cells that require a process called the progenitor cell intermediary. Although considered as the safest and the cleanest way to perform gene therapy, it is rather labor-intensive. Reprogramming is the addition of one or more genes to cells to facilitate a change in the cell's characteristics. Cell elimination, on the other hand, is considered as making the cells kill themselves or suicide to destroy the cell. That is used to destroy malignant or cancerous tumor cells and even the overgrowth of benign or non-cancerous tumor cells, in which these cells are eradicated by the means of releasing suicide genes that target the said cells to let go of a prodrug that kills the tumor cells.
What are the uses of gene therapy? Gene therapy treatment modalities are currently used for the treatment of rare diseases, different types of cancers, and other diseases where alternative or far superior treatment options are non-existent. Gene therapy has shown potential to cure a spectrum of ailments by preventing the development and alleviating symptoms of uncommon and sometimes life-threatening ailments. Adenosine deaminase deficiency, severe combined immune deficiency, and other immunological deficiencies are among those that can be effectively addressed using gene therapy. Another illness that can be treated with gene therapy is inherited blindness. In the case of Loxterna, a copy of RPE65 gene is delivered to the patient's eye using a viral vector, and the virus modified to eliminate its capacity for infection and introduce a functional gene that can be used by the patient's cell to generate a functional protein. Another illness that can be treated with gene therapy is inherited blindness. In the case of Loxterna, a copy of RPE65 gene is delivered to the patient's eye using a viral vector, and the virus modified to eliminate its capacity for infection and introduce a functional gene that can be used by the patient's cell to generate a functional protein. Many potential cancer gene therapies are now being tested, and many have had full remission. Gene therapy transfer technology for cancer treatment holds a great promise for increasing the effectiveness of current chemotherapeutic treatment regimens. Significant advances have been made in the field of oncolytic virotherapy, and trials are in progress that incorporate this technique for brain cancers as well as cancerous treatment. Gene therapy has improved the ability of Parkinson's disease patients to control their muscles. A gene for GAD was introduced into their subthalamic nucleus in the brains of the patients with PD. The goal was to increase the presence of GABA in the brain area, thereby helping reset the abnormal circuitry of the Parkinson's brain. The following table summarizes gene therapy products that have acquired full market authorization and is currently available in the market.
What are the common types of cell therapy? The most common type of cell therapy is blood transfusion. Blood transfusion is a medical term which means a procedure used to transfer blood or some products based on blood, such as white blood cells, red blood cells, and platelets from the circulatory system of one human to that of another human. Another common cell therapy is the transplantation of hematopoietic stem cells to create bone marrow, which have been performed for over 40 years.
Viruses are incredibly good at delivering nucleic acids to specific cell types, while avoiding immune surveillance by infected hosts. Because of these characteristics, viruses are appealing as gene delivery vehicles or vectors for gene therapy. Several viruses, including retroviruses, adenoviruses, adeno-associated viruses, and herpes simplex viruses, have been genetically modified for use in gene therapy applications. Because each of these vector systems has distinct advantages and limitations, each has applications that are best suited to it. Retroviral vectors can permanently integrate into the genome of an infected cell, but transduction requires a mitotic cell division. Adenoviral vectors can efficiently deliver genes to a wide range of non-dividing and dividing cell types, but infected cells are eliminated by the immune system, thus limiting the gene expression in vivo. Adeno-associated viruses, or AAV, can also infect both dividing and non-dividing cells. However, it has a limited DNA capacity. Lastly, herpes simplex virus can deliver large amounts of exogenous DNA. However, cytotoxicity and transgene expression luminance remains a challenge.
No, because these viruses used in gene therapy are designed to reduce the risk of adverse effects. These viral vectors are tested in animals and in human trials to make sure that human cells are not harmed. However, like all treatments or therapy, it has its risk. Therefore, availing of this therapy must discuss with their physicians and other health advocates about the pros and cons of the gene therapy.